Imagine a virus so elusive, so quiet in its early moments, that it can traverse continents before the world even notices. The Nipah virus, NIP, belongs to a family of viruses that challenge our understanding of how microscopic life interacts with us. Its origins trace back to fruit bats, creatures that have coexisted with humans for millennia, silently harboring pathogens we are only beginning to comprehend. When these viruses leap from bats to humans or intermediary hosts like pigs, they illuminate the fragile threads connecting our ecosystems, our agriculture, and our health. The origin of the Nipah virus traces back to one of nature's most fascinating and misunderstood creatures, the fruit bat, or flying fox, which quietly roams forests and orchards, bridging ecosystems without ever fully revealing its hidden microbiome. These bats harbor the virus as part of a delicate balance in their own bodies, coexisting with pathogens in a way that humans rarely achieve, a coexistence shaped over millennia of evolution. Their role as reservoirs is not born from malice or randomness, but from an intricate web of ecological interactions where the virus persists without necessarily causing disease in its natural host. When humans enter their habitats, whether through deforestation, agricultural expansion, or urban encroachment, the barrier between species thins, creating opportunities for the virus to cross into new hosts. In some regions, pigs act as intermediary hosts, amplifying the virus silently within densely populated farms where humans are in constant proximity. The virus's leap from bat to pig and eventually to human is a reminder of the fragile interface between wild ecosystems and human society, a threshold where microscopic events can reshape the health of entire communities. These initial transmissions are often subtle, unnoticed in their earliest stages, and yet they hold the potential to ignite outbreaks that ripple through villages, towns, and sometimes entire regions. Understanding this origin requires more than just identifying a host species. It demands an awareness of the environmental pressures and human behaviors that facilitate zoonotic spillover. Climate variations, habitat destruction, and the seasonal patterns of fruiting trees all influence bat behavior, determining when and where the virus is most likely to encounter new hosts. Observing these patterns is a window into the broader dynamics of life on Earth, revealing how interconnected our health is with the rhythms of the natural world. Scientific investigations into the origin of Nipah have not only mapped the presence of the virus in bat populations across South and Southeast Asia, but also illuminated the subtleties of viral adaptation, how it evolves to exploit new hosts while maintaining its viability in a reservoir species. This evolutionary resilience underscores the importance of surveillance and ecological understanding. Knowing where the virus resides and how it moves allows us to anticipate risk and implement interventions before human infection occurs. Yet beyond the laboratory data and ecological models, there is a profound lesson in reflection. The emergence of Nipah is a signal, a mirror reflecting the consequences of our interaction with nature. It prompts consideration of the ethical and practical responsibilities we bear, as human activity increasingly encroaches on habitats that were once remote, creating conditions for viruses to cross species boundaries. The story of Nipah's origin is therefore not just a tale of a virus, but a meditation on the interdependence of life, the resilience of certain species, and the delicate thresholds at which human and natural worlds intersect, offering insight into how microscopic agents can illuminate the vast and intricate connections that sustain life. The spread of the Nipah virus reveals both the fragility and the remarkable complexity of the networks that connect human life. It is not a pathogen that moves recklessly, but one that exploits proximity, intimacy, and the rhythms of daily existence, quietly traveling along invisible pathways between bodies. Human-to-human transmission occurs primarily through close contact, particularly when individuals care for the sick, exposing themselves to respiratory droplets, saliva, urine, or other bodily fluids. In hospitals and households, the virus can move from person to person almost imperceptibly, a reminder of how interconnected our lives are even in moments of solitude. Contaminated surfaces, objects that carry traces of viral particles, serve as silent intermediaries, linking individuals who may never directly meet, yet who share a space and time in the same environment. In agricultural settings, the virus can amplify through intermediary hosts such as pigs, whose dense populations and frequent contact with humans provide the perfect bridge between wild reservoirs and human communities. Seasonal and ecological factors influence these dynamics as well. Fruiting seasons attract bats to orchards, increasing the likelihood of contamination of food sources such as date palm sap or fallen fruit, which may then be handled or consumed by humans, creating a subtle yet potent chain of transmission. The virus's spread is not random but shaped by patterns of human behavior, cultural practices, and environmental conditions, highlighting the delicate interplay between biology and society. Observing how Nipah moves teaches us about the invisible contours of contagion, the thresholds where hygiene, distance, and protective measures can either halt or accelerate its progress. It underscores the necessity of early detection, isolation, and community awareness, yet also invites reflection on the ethical responsibility of caring for the sick while protecting oneself and others. Each transmission event is a quiet dialogue between the virus and its environment, between human activity and natural reservoirs, reminding us that outbreaks are often symptoms of larger ecological and societal imbalances. By tracing these pathways, scientists gain insight not only into the mechanics of viral spread, but also into the human behaviors and environmental pressures that make such spread possible. The story of Nipah's transmission is therefore more than epidemiology. It is a meditation on vulnerability, connection, and the unseen forces that shape health across individuals, families, and communities. Understanding its movement allows us to navigate these forces with both practical caution and reflective awareness, appreciating that every act of prevention, every moment of hygiene, distance, or mindful interaction, is a step in the intricate dance of coexistence with the microscopic world. The signs and symptoms of Nipah virus infection are deceptively subtle at first, a quiet whisper of imbalance within the body that can easily be mistaken for ordinary illness. In the initial days, fever and headache emerge, accompanied by fatigue and generalized weakness, signals that the body's immune system has sensed a foreign presence and begun its complex orchestration of defense. These early indicators often pass under the radar, blending with the common ailments that punctuate human life, yet beneath them, the virus is already interacting with host cells, infiltrating tissues, and silently challenging the nervous system. As the infection progresses, the body may reveal its struggle more dramatically. Respiratory difficulties can arise, with coughing or shortness of breath reflecting inflammation within the lungs, a delicate organ system that balances oxygen intake with the precision of countless microscopic processes. In some individuals, neurological symptoms develop, subtle at first with confusion, disorientation, or drowsiness, then potentially escalating to seizures, severe encephalitis, or a diminished level of consciousness. The virus's ability to target the central nervous system is particularly striking, a reminder of how microscopic agents can exploit vulnerabilities in the body's most complex structures, influencing thought, perception, and control with a precision that is both humbling and terrifying. These manifestations are not uniform. Each human body responds differently, shaped by genetics, immune function, and overall health, which is why early detection and careful observation are critical. Even minor symptoms should never be dismissed, as they may signal the beginning of a cascade that can accelerate rapidly. The subtlety of early signs underscores the importance of vigilance in communities at risk, of listening to the body and to the patterns of illness that emerge in those around us. Observing the symptoms is not just a clinical exercise, it is an invitation to recognize the body's inherent wisdom and fragility, to appreciate how closely health is intertwined with the unseen world of microbes. By understanding these early and later signs, scientists, healthcare workers, and caregivers gain insight into the virus's behavior and the body's response, creating opportunities to intervene, support, and protect. Each symptom becomes a narrative of interaction, a story of microscopic conflict and cellular resilience, reminding us that disease is not only a biological event but a profound human experience, reflecting the delicate balance between vulnerability, awareness, and the pursuit of healing. Prevention of Nipah virus is both a practical necessity and a profound exercise in mindfulness, a way of acknowledging the intricate ways human life intersects with nature and microbial worlds. Because the virus often emerges from fruit bats and intermediary hosts like pigs, one of the most effective strategies is reducing direct exposure to these animals and their secretions, a practice that requires awareness of daily behaviors that may seem ordinary but carry hidden risk. Consuming raw date palm sap, for example, or handling fruits that have been partially eaten or contaminated by bats can become a pathway for the virus to cross species, a subtle reminder that human sustenance and the natural environment are deeply intertwined. Maintaining strict hygiene, washing hands frequently, and disinfecting surfaces in homes and hospitals is more than a routine, it is a conscious act that interrupts the invisible chain of transmission, protecting not only the individual but the broader community. In agricultural settings, careful handling of livestock, separation of species, and monitoring animal health are essential, recognizing that pigs and other animals can amplify the virus silently before humans are ever aware. Community awareness and education are equally vital, as prevention is as much a social and cultural endeavor as it is a medical one. When families and neighbors understand the risks and the measures that can be taken, they collectively create a buffer against the virus's spread, transforming knowledge into a shield that extends beyond personal boundaries. Seasonal and environmental awareness also plays a role, understanding the movement patterns of fruit bats, the timing of fruiting trees, and other ecological factors can guide behaviors in ways that minimize risk without severing our connection to nature. Personal protective measures, such as masks or gloves in high-risk settings, become acts of both caution and care, embodying a recognition of vulnerability without succumbing to fear. Prevention, in this sense, is a philosophy as much as a set of practices. It is a way of living that honors the delicate balance between humans, animals, and the microscopic entities that share our world. Each deliberate action, from washing hands to monitoring animal health, is a small but meaningful contribution to collective resilience, a reflection of the human capacity to anticipate, adapt, and coexist safely with forces we cannot fully control. By embracing prevention thoughtfully, we engage in a dialogue with the unseen, respecting the boundaries between species while cultivating a sense of shared responsibility and foresight. Testing and treatment of Nipah virus are windows into both the precision of modern medicine and the patience required when facing an elusive pathogen. Laboratory detection, particularly through PCR testing, allows scientists to identify the virus at its earliest stages, even before the full spectrum of symptoms has appeared, offering a critical opportunity to intervene and limit its spread. These tests are not merely technical procedures, they are instruments of foresight, translating microscopic traces into actionable knowledge, guiding healthcare providers in the delicate decisions of isolation, monitoring, and supportive care. The process requires skill, timing, and coordination, reflecting how human ingenuity can illuminate otherwise invisible threats. Treatment, however, remains largely supportive, a careful orchestration of maintaining vital functions while the immune system confronts the virus's presence. Clinicians monitor respiratory function, fluid balance, and neurological status, intervening as needed to sustain the body's natural defenses, a reminder that in some battles, medicine serves as a guide rather than a cure. Research continues into antivirals and potential vaccines, driven by an understanding that prevention and preparedness must coexist with the quest for therapeutic solutions. Each intervention, whether a diagnostic test, a ventilator, or experimental therapy, is a negotiation between knowledge, technology, and the body's innate capacity for healing. The challenges of treatment also illuminate the ethical dimensions of care, emphasizing compassion, vigilance, and the responsibility to protect both individual patients and the broader community from further transmission. Observing and managing Nipah infections teaches humility, highlighting that even in an age of advanced science, patience, observation, and respect for the body's complexity are essential. It underscores the interdependence of medical innovation, public health strategies, and individual resilience, demonstrating that fighting a virus is not simply a matter of eradicating a pathogen but of navigating the intricate interactions between biology, behavior, and environment. In this light, tests and treatment are more than clinical tools. They are mirrors reflecting our understanding of life at the microscopic scale, of how disease emerges, spreads, and can be contained, and of the continuous dialogue between human knowledge and nature's subtle forces. Each diagnostic decision, each supportive measure, each research endeavor contributes to a growing tapestry of preparedness, reminding us that confronting a virus like Nipah is as much about insight, foresight, and reflection as it is about medicine itself.